So we'll
Um,
talk about hyperpolarized magnetic resonance of exchangeable protons.
yes, so first of all, thank you for organizing this beautiful conference. I think one of the younger scientists, and I find it very nice. I learned a lot, and I'm also very glad that I can present my own research. So it's indeed about the hyperpolarization of exchangeable protons. So exchangeable proton pool using parahydrogen. So why the hyperpolarization of a bulk solvent or a proton pool, exchangeable proton pool? If you can hyperpolarize this, you have advantage of using this bulk hyperpolarization and using chemical exchange or, for example, Noye effects to hyperpolarize secondary targets. Now, the most famous and still the most effective procedure of hyperpolarizing a source of bulk exchangeable protons is dissolution DNP. So you hyperpolarize a tiny batch of water at low temperatures and irradiate with microwave radiation, and then you send it to high field, and then you make contact with the secondary targets, in this case proteins with amide or amide bonds. Now, indeed, we are a parahydrogen conference, so you have heard it a lot. The hardware of doing this is quite expensive, and the conditions when you are accomplishing this are also very harsh. So there are also a handful of papers, and I think my host is one of them. So Professor Bowers already mentioned this in hyperpolarizing exchangeable protons or the exchangeable protons of, for example, methanol or water itself, and trying to do this with parahydrogen, in which I'm also going to discuss this. And also the hardware needed for this is quite inexpensive, and the production of parahydrogen is easy. Now, so indeed a handful of papers. Some of the papers use homogeneous catalysis, so the famous N-heterocyclic carbene iridium catalyst, so with the IMAS ligand on top. Some papers have published results in hyperpolarizing, so this is bulk solvent. And also, of course, the beautiful talk already in the previous session of Zhao from the Bowers group has beautifully shown the platinum-3 tin alloy disclosed by mesoporous silica and by surface catalysis, trying to do the same thing, so hyperpolarizing this water, this bulk solvent. Now, what I'm going to discuss today is this topic. So with still homogeneous catalysis in methanol system, trying to hyperpolarize this exchangeable proton source. Now, some of you may see the equivalence with this also beautiful research, mostly centered in the Duckert group about cyber relay. The only difference is I'm only going to speak about systems in Prote conditions, so with a lot of exchangeable stuff. And in their research, it's mostly based on aprotic conditions, so working in, for example, dichloromethane, deuterated dichloromethane. Now, so what we actually, by coincidence, seen, so we use this special kind of molecule. It's an aminopropyl dietoxymethylsilane. So you have the aminopropyl group over here with the silicon center metal group and two etoxy groups. If we combine this with the traditional iridium IMAS catalyst, so this is the carbene catalyst, cyclooctadiene, contacted with parahydrogen, in some case added ammonium carbonate buffer. We bubbled at low field, so I will explain the hardware later, but bubbling at low field, transferring to high field, what we noticed, instead of actually what we are trying to see in the past, we wanted to see hyperpolarization of silicon itself, but what we noticed is that this system, somehow at room temperature, was very efficiently in hyperpolarizing the exchangeable protons of, in this case, methanol, which is not that easy to accomplish, but what we saw was incredible, and we wanted to investigate why this was happening. So why this molecule itself? Why is this worth investigating? One of the things is its ability to indeed hyperpolarize at room temperature these exchangeable protons. You will see later also it tends to have a strong interaction with iridium, which is not that really expected because an amine is not that interactive with iridium compared to, for example, an cycle, so pyridine. One of the very good things is it has a huge grafting potential. So we are now researching what the grafting potential is. And so what you can do is these epoxy groups are, they can go off. So, and you can displace them by, for example, a cyanol group. So you can graft this ligand on a silica surface, which is quite amazing. And then also, so the goals also later trying to expand to an aqueous solution. So instead of using the IMS ligands and trying to, as well as the publication of Leuven itself, trying to see if we can exchange the IMS for a water soluble system, and then see if we see the same effect also at room temperature. Now, first I'm going to discuss a little bit about mainly the low field dependence of this hyperpolarization here. Also discuss how we managed to see the interaction between this aminosilane and iridium, and then afterwards a general, or trying to come up with a mechanism of why this is happening in our system. First of all, so the experimental setup. So we were very lucky to collaborate with Brugger on using this system here. So it's a prototype field cycling device in which we bubble our solution. So we administrate Parahydrogen to our solution, and then we can bubble and then transfer to high fields. And this, so high field is here 800 megahertz, and this takes about 2 seconds. In which we bubble 2 minutes, so we activate 2 minutes at low field, and then we can also change the low field value. We go to high field, and then we measure. So this total sequence takes about 2 seconds after the bubbling stopped, and these are the other concentrations of catalyst we use. Now, there is already been some discussion about whether to use a shaking method, or why for example, the FRIT, this system would not work that well. We haven't seen any experience. So for me, this setup is incredibly to use, to have a reproducible system to test timings and to see if, for example, different Gaussian fields have different effects. So for me, this is a beautiful system to work with. So as you can see here, one of the first things we tend to investigate was which dependency on which field has this exchangeable proton hyperpolarization. And as you notice that immediately, it seems to have a Sabre type of correlation in which at the typical fields, also, for example, seen in pyridine, around 7 millitesla, plus and minus 7 millitesla, we see the maximum in intensity, where spin order is transferred from Parahydrogen, effectively in this case to the hydroxyl groups. What we didn't expect is actually the no correlation between, or no dependency on the hydrogen hyperpolarization, which we definitely saw, and also the hydrides. I will say a little bit more about this because there are quite a few. Normally in a pyridine system, you should expect a mirror image of the hydroxyl. So at this 7 millitesla, it should be a little bit higher, and in this case, we didn't see anything. So to take in mind the Sabre type behavior, it's important for later explaining the mechanism. Now, what is nice, when you have some quantitative data on prepolarization field dependency, you can just see, so one data point is actually one measurement with one field value, and you can use how much, for example, hydrogen and hydride is hyperpolarized, and as you can see, they are quite correlated. So if we see hydrogen of the hydrogen hyperpolarization, they tend to be hyperpolarized, and it is just to the already discussed chemical exchange between hydrides on the catalyst taking hyperpolarization and then off. And this is a beautiful graph showing that it's indeed perfectly correlated in our system too. Now, one of the things we had difficulties with, we used quite an amount of concentration of this ligand, and we had difficulties to see, or we just actually didn't see any attached ligand form of this to our iridium system. So in a pyridine system, you can easily distinguish which pyridine is bound if it's hyperpolarized. In our case, we didn't even see hyperpolarization of this molecule here. So what we tried to do, we wanted to make these amine protons visible, and then we worked in apotic conditions in which we immediately saw that the NH2 resonance, as you can see here, so the amine resonance is indeed hyperpolarized, which concludes that this is an expected binding position of this big molecule, or a little bit bigger molecule than normal on the iridium. So what we did next, we couldn't see any of the aminosilane resonances bind it. So what we did indirectly, we checked how this carbene ligand here changed upon introduction of aminosilane into our system. So this is the aromatic section of the IMS spectra. This is the aliphatic section in which you can see all the metal groups. And what we did see, if we introduced this aminosilane ligand, we immediately saw broadening of these resonances here. So these metal groups, they broadened upon addition, which is normal, because if we attach a bigger ligand, so the steric hindrance will broaden these IMS molecules, so these metal groups here and also the off-axis groups. So we did see expected binding towards this catalyst over here. Next step, administration of Parahydrogen. And what was noticed, similar to a normal pyridine system, so this is one possible form of how our catalyst looks like at the end. We did see, if you use Parahydrogen and you bind, you make a more open system, so octahedral 18 electron complex, and you definitely see this in the IMS molecules, so the metal groups which are sterically hindered when in this kind of configuration, and when you have an open system, 18 electron, they can move through this axis, and you have no discrepancy anymore between H1 and H5. So this was an indirect tightening of that. Indeed, aminosilane is quite good in interacting with our catalyst. Now the hydrides, as you may expect, if you only have this catalyst in solution, or this type of catalyst, you only should expect one resonance. So you have a symmetric system. We have multiple, which we are explaining because we use the deuterated solvent in which you can make multiple amine functionalities, in which also these can shift. What we did also see, so it's not shown here, but the data is there, perfectly visible. We have exchange, so iridium also catalyzes the etoxy groups going off. So this is also why it is a craftable ligand. We have exchange from methanol and ethanol going off, which makes our ligand actually variable over time, in which this also changes. And then also the most important thing we tried to investigate was silicon itself. Is it interacting with our iridium? It's known that silicon and nitrogen, you can make binded ligate pincers with this. And to investigate this, we tried a chemical because we didn't see any silicon itself. So what we tried to do chemically is actually investigate if silicon was important just by omitting any silicon functionality in our product. So we tried exactly the same measurements, bubbling at low fields, transferring to high fields, but with an easy amine group, methylamine. And indeed, you see a much lesser extent of this exchangeable proton hyperpolarization, which I will explain the mechanism or what we think the mechanism is in a bit. But this is one of the first steps in maybe seeing indeed, is the silicon atom important? Later experiments which really should amplify this result is the use of cyber sheet and micro Tesla prepolarization fields to see if indeed silicon was binding. It should also transfer some hyperpolarization to the silicon, but in our setup we still cannot see this. The mechanism itself, so the first steps, as you noticed, so we have the NH2 hyperpolarization, we have the cyber field dependency, we have the monitoring of APDMS interacting with our ligands. So this is a sort of first effect of what is happening. The only step is this one, the uncharacterized activated complex. So we know we will have some hyperpolarization from the parahydrogen going through the NH2 bonds. And then afterwards, how is the exchangeable proton itself hyperpolarized? So it's based on a paper published some 5 years ago. But indeed, what is happening? We have quite an amount of A+ PDMS in our system, which is actually giving its protons. So this cationic species, quite important, is giving their extra protons to the hyperpolarized APDMS going off the ligands, making A+ PDMS and then transferring it to the solvent itself and making an exchangeable proton pool, which you see effectively on the spectrum. Now, why APDMS itself and not the other ligands? APDMS is a quite basic molecule. So this is measured in water and also not deuterated water, but they are similar. So these values will be similar in a deuterated methanol system in which this reaction, acid-base reaction, is going. So what we have quite an amount of A+ PDMS in our system. It's even 25%, in which it seems that the equilibrium between a PDMS itself, which can ligate with the catalyst, and a plus PDM which cannot ligate because it's a positively charged molecule and also a positively charged catalyst. This equilibrium seems to be quite important. So you can ask yourself, is this possible also with other molecules? Why, for example, is pyridine not showing this in a normal experiment? This was explained 5 years ago by Moreno, a beautiful publication. In a normal situation, pyridine, as you can see, the difference in pKb from 4 to 8.77 is quite high. So this pyridine or pyridinium ion, PER plus, is it shown here? It's not that large in the system. So what these guys did is, what if we acidify the system? So they did it with aqueous HCl solution. What if you acidify this, you make this quantity much bigger. And what they indeed see, with some different extent of Gaussian fields, they saw indeed a positive hyperpolarization in that case of their HDO CD3OH system. Now, to really counter the effect of water itself, because if you add an acid dissolved in water, water itself can also interact with iridium. In our case, if we added water, we still saw the same effect, but to prove that it was not water doing any strange things here, we tried to mimic the same experiment, but instead of adding HCl in an aqueous solution, we tried to add indirectly anhydride, a tiny amount of anhydride, which dissociates to acetic acid, so without any water in it, and indeed making the pyridinium concentration much higher in our system. And what we expected to see, and what we indeed did see, is if you have an amount or a higher amount of this pyridine plus concentration, you indeed see hyperpolarization. It is a red one, and then after more and more scans, after 10 seconds, you see it indeed going down, pyridine the same, so from negative to positive. So this is one of the examples of experiments that indeed unify the mechanism that these cationic species are quite important in hyperpolarization. So here you even see so this PNL effect, and it also shows, so this is also where I want to conclude this talk. It also shows that APDMS itself, these properties, they allow for this negative hyperpolarization of exchangeable protons in quite general conditions. You don't have to acidify, you don't need to add any acid. You can do it at room temperature. And we discussed the interaction with the iridium transition metal, which is quite strong for this untypical amine. We've shown this indirectly through the IMS ligand, and also together with the low field dependency that has a Saber type characteristic. And also it seems to be the importance of the cationic A plus PDMS and also pyridinium ion. It seems we have a sort of maybe already some clues about what is important, and also we can base our research further on trying to getting the system more efficient. Future prospects, of course, is the water hyperpolarization itself. So one of the things we can try is to graft both our APDMS and our ligand and our catalyst to silicon support, sorry, and then see if we can do the same in aqueous conditions by changing our catalyst a little bit and see if we see the same effect. And of course, the most important step is trying to use this source of exchangeable protons for secondary target hyperpolarization. So with this, I think I said everything, and I want to thank again our research group here, and also the collaboration with Broeker, David and Jean-Max and James really have helped me in doing this exciting research, and I am open to any questions.
Wow, thank you very much. That was quite an extensive study you did right there. And I will try to sum up the questions as best as I can. So first of all, someone writes, I have a question. I think you said you add an ammonium buffer to these experiments.
Yes.
Ammonium is not coordinating itself and contributing to the relayed proton exchange effects.
So that's true. That's true. So what we didn't see much change between if we didn't apply it. So this ammonium carbonate. So it's over here. If we didn't apply this, we still saw the same effect. So we saw it in a lesser extent. So for example, if we included it, it was 2 or 3 times higher, which is already some extent. But we figured out that indeed this can coordinate it. If it should coordinate, it already has to make ammonia from a salt. But why is this also important? Because in a system with a lot of deuterium ions, you have the negative effect of that as you can see here, that you have deuterium scrambling, and this type of amine cannot introduce any hyperpolarization because these deuterons cannot be hyperpolarized. If we add a little bit extra protons from this also amine ammonium carbonate buffer, you indeed include, first of all, an extra source of making of trying to equilibrate the difference, because it's a buffer between NH, so A plus PDMS and A PDMS, and also add a little bit more protons itself, which can be hyperpolarized. So we indeed think of this, and it can be that it still makes a little bit of maybe indeed NH3 ligands here. You still have to investigate that, but it's quite interesting fact already. Just to add, because I don't think it was by coincidence that we've added this for buffering our system, but indeed it gives some additional results, and it's indeed quite interesting to further investigate what definitely more in detail what this guy is doing and why it is enhancing our result over here.
Okay. Well, on a similar thought or subject about the mechanism, question: have you ruled out a 1H type mechanism?
Can you 1H FIP?
1H FIP type, yes.
Yeah. So one of the things where we based our results on were the really same or cyber type of hyperpolarization we get here. For 1H FIP, we should see, because I based mainly, so a lot of my research was based on the paper of 2015 from Moreno, paper that you actually wrote, and you saw much more different Gaussian field dependency, just much higher, because you used, I think, histidine.
Yes.
But one of the things we ruled out, you see it here, for 1H FIP, we thought maybe if we used for example, if we really used the hydride itself, so the hydrogen itself, we should indeed see HD formation, which indeed we see, because it's a result shown in the publication itself. But what we did see, this was just happening. So this is a static measurement with our solution, but at high field. So it was no Gaussian field or low field. It's just at high field monitoring what is happening to our hydrogen and also what is happening to our HD. Indeed, we have some situation where HD is formed. So these are some graphs where our octahydrogen is going down and HD is forming. But we do think it's an independent process. It's just another side process that is happening, because we have this measurements, this typical cyber type measurements of Gaussian field dependency, and also the very similar approach. Why it is negative, it's still not, because this is very similar, but they have positive hyperpolarization, and in our case it's negative. That is still a big question to solve. Why in our system is it negative and in theirs positive? But there are some exciting things to investigate to come to a sort of united system in the hyperpolarization of exchangeable solvents in general.
Yeah, and maybe both things are going on. So if you study at higher fields, then because it can happen. So on to another question, because so many people ask about applications. You already have shown polarizing such a sort of big molecule with silicon in it. You think macromolecules will be possible one day?
One of the things we indeed see, so we need to see quite a big molecule being attached to our ligand. That I think that's for sure what we proved in the IMS behavior and the NH2. But what we somehow, our APDMS is so special, it doesn't transfer any hyperpolarization to these carbons, to these protons. So in our normal spectrum, they are just a little bit slightly down, but we accounted this to, if you transfer your molecule, you transfer your solution so fast from low to high field, you have the opposite relaxation effect, that just your system itself is not fully equilibrated, and you have a slightly negative or less positive polarization of this peak. So we didn't see any, definitely not negative hyperpolarization of these peaks. we only saw hyperpolarization on H2 and then transferring it efficiently to the other protons in general, which is a nice result. But I don't think it mainly will be the next step is trying to see if we can transfer from this pool we have, transfer it further to other molecules. And if these are big enough or bigger, and they have a lot of exchangeable protons, maybe it is possible.
Very exciting. Thank you so much for your talk and your extensive study. Sadly, we're running out of time, so all other questions have to go on Slack.
No problem.
And I'll try to get our next speaker ready here.
Okay, thank you.
Ah.